Hello friends, welcome back to my YouTube channel Nesting Padasala. This is a part 2 video. Currently we are revising for our BARC examination. Which of the following represents an autonomous retrotransposon? The correct answer is line. Line is an autonomous retrotransposon. They have their own reverse transcriptase enzymatic activity. Whereas the sines, they are non-autonomous retrotransposon. They don't have the reverse transcriptase activity. Example of lines include L1 family and example of sines include ALU element. Both lines and sines are highly present in human. Regarding to other transposon like ACDC element is present in maize and P element and hobo element are from Drosophila and TY element, Copia and Gypsy is also from Drosophila. Copia and Gypsy will be coming in a classification called retrotransposon and the piggyback is from moth and sleeping beauty is from salmon. Following are some of the characteristics of MSC class 1 and MSC class 2 molecule except one which is applicable only for MSC class 1. Okay, so the correct answer is MSC class 1 only. They will be expressed on all nucleated cells. Okay, whereas MSC class 2 will be present on all cells, that is antigen presenting cell. Regarding the structure of MSC class 1, they have alpha chain and beta 2 microglobulin, and regarding to MSC class 2, they have alpha chain and beta chain. Regarding to peptide binding, MSC class 1 will be process the endogenous antigen and MSC class 2 will be processing an exogenous antigen. And peptide binding groove in MSC class 1 will be formed by alpha 1 subunit and alpha 2 subunit, whereas MSC class 2 the peptide binding groove is formed by alpha 1 subunit and beta 1 subunit. So MSC class 1, the size of the peptide should be ranging from 8 to 9 amino acid long, whereas for MSC class 2, the size of the peptide should be ranging from 12 to 17 amino acid long. Okay. The next is cytotoxic T cell expressor. Cytotoxic T cell, they will be expressing a CD8 marker and they are called class 1 MSC restricted. Next, the melting temperature of DNA is higher when the content of GC base pair is higher. Okay, so for those DNA sequence which are having a high amount of GC base pair means ultimately the time taken to melt half of the DNA will be definitely higher because there will be 3 hydrogen bond present between G and C. Next is in eukaryotes, nucleosome comprise of which of the following histone? Okay, with regarding to nucleosome, it is made up of H2A, H2B, H3, H4. There will be 2 subunit of H2A, there will be 2 subunit of H2B, there will be 2 H3 and there will be 2 H4. Thereby they will be forming 8 histomer, that is 8 histone molecule together they will be forming a octamer. Okay, so one nucleosome will be connected to another nucleosome by a linker DNA which is called as linker histone and this H1 is the linker histone. Okay, whereas H2A, H2B, H3, H4 they are present as a dimer and they will be forming a core histone. Okay. Next is during signal transduction on one molecule of phosphorylated inositol 4,5 bisphosphate is cleaved into one molecule of each inositol triphosphate and diacylglycerol by which enzyme? So the correct enzyme is phospholipase C. Okay, so this inositol triphosphate they are soluble and they will be entering and they will be signaling the endoplasmic reticulum to produce more amount of albumin. This diacylglycerol they are hydrophobic, so they will be present in the plasma membrane only. Okay, so you should know the enzyme responsible for cleaving both the IP3 and DAG is with regard to phospholipase C. Eukaryotic RNA polymerase 2 transcribe. Okay, so here I had given RNA polymerase will be transcribing your ribosomal RNA like 28S, 5.8S, 18S. Whereas RNA polymerase 2 is involved in transcribing messenger RNA and SM family of small nuclear RNA and some nuclear RNA, siRNA, miRNA, PV interacting RNA and long non-coding RNA. Whereas this RNA polymerase 3 is involving in transcribing your transfer RNA, 5S ribosomal RNA, LSM family of small nuclear RNA and other small nucleolar RNA, 7SL RNA and 7SK RNA. Okay. So here the question is that eukaryotic RNA polymerase will be transcribing. So it will be transcribing your all your small RNA, small interfering RNA, micro RNA and PV interacting RNA and messenger RNA. Okay. So the correct answer for this particular question is can able to say messenger RNA, micro RNA, small nuclear RNA or the RNA which are transcribed by RNA polymerase 2. Okay, whereas your transfer RNA is transcribed by RNA polymerase 3 and your ribosomal RNA is transcribed by RNA polymerase 1. So only for 28S, 5.8, 18S ribosomal RNA is transcribed by RNA polymerase 1 and regarding to 5S ribosomal RNA it is transcribed by RNA polymerase 3. Which enzyme will be digesting RNA in DNA-RNA hybrid? Okay, the correct answer is RNA sketch. So the sketch is for hybrid. Okay, so RNase 1 is from Saccharomyces cerevisiae. They will be cleaving at 3' end of all your single-strand RNA. And RNase 2 is also responsible for cleaving single-strand RNA. And this RNase 3, which is isolated from E. coli, Saccharomyces cerevisiae and Saccharomyces bombay. Okay, so this RNase 3 will be cleaving a double-stranded RNA. Okay, so this RNase 3 is involving in processing of your ribosomal RNA. Okay, so next is RNase P. So this RNase P is responsible for cleaving an extra sequence in your tRNA molecule. So for the post-translation modification of tRNA, this RNase P will be engaged. Okay, next is RNase A is isolated from bovine pancreas. It will be hydrolyzing your phosphodiester bond in your RNA that will specifically cleaves after either cytosine nucleotide base or uracil. Okay, it has a capacity to cleave only in pyrimidine base pairs. Okay, regarding the RNA sketch, they are involving cleaving your RNA present in RNA DNA duplex. That is, during your lagging strand synthesis of your DNA, this RNA-H will be cleaving the primer and the DNA sequence. Which of the following immunoglobulin cause type 1 allergic reaction? So allergic reaction means definitely you should know it is IgE. Next, which of the following regions of genome are best suited for generating DNA barcode for species identification? So for DNA barcoding studies, they will be generally taking organelle genome or else they will be taking ribosomal genes. Okay. So in animals, these are the list of marker genes used for DNA barcoding. This includes cytochrome C oxidase gene and 12S ribosomal RNA and 16S ribosomal RNA. Okay. In plants, the most commonly employed marker for DNA barcoding includes methylase kinase, MATK, or Rubisco, and photosynthetic genes and ITS. Whereas in bacteria, the commonly employed marker for DNA barcoding include RPOB. Okay, which is RNA polymerase subunit B. In fungi, they will be generally employing internal transcribe spacer region, and in protists also they will be generally employing ITS. Okay, these are the markers that are used for barcoding. So generally they will be taking the genes from organelle DNA. Okay, so the organelle DNA genes will be acting as a best marker for DNA barcoding studies. Okay, because in mitochondrial DNA there is no intron is present, and this mitochondrial inheritor they will be present in high amount. They are abundant. That's the reason why they are generally taking the genes from the mitochondrial DNA in case of animals, and in plant they will be taking the genes from chloroplast. Okay, L-tyrosine serves as a precursor for. So this L-tyrosine they will be serving as a precursor for dopamine, norepinephrine and epinephrine. Whereas the serotonin will be acting as a serotonin will be produced from the tryptophan. Okay, next the amino acid like alpha ketoglutarate is responsible for producing glutamate, and from the glutamate your GABA, that is gamma aminobutyric acid, will be produced. Okay, with regarding to catecholamines, the responsible precursor is tyrosine. Tyrosine will be converted into dihydroxyphenylalanine, and dopa will be converted into dopamine, and from dopamine, in the presence of dopamine beta hydroxylase, norepinephrine will be produced, and from norepinephrine, epinephrine will be produced. The repeat sequence of telomere in humans is, the repeat sequence of telomere region is TTA-GGG. Okay. So the next is with regarding to the cytosine residues in DNA or methylated specific di or trinucleotide sequence. Which are the following methylation motifs is predominantly found in case of protein coding genes in animal? Ultimately, it is CpG island. Whereas in plant, the primary site for methylation activity is CHG or CHH. The H might be either adenine, thymine or cytosine. Next, cotomorphogenesis is characterized by, so this cotomorphogenesis means germination under light or germination in the presence of dark. Okay, so when the plant is germinating in the presence of dark means it will be having a small cotyledon and it will be having a closed cotyledon and they have a elongated hypocortical. Okay, so next, which one of the following contributes to the development of male reproductive system? Ultimately, it is anti-Mullerian hormone AMS is responsible for development of male hormone. Okay, so here you can able to see till 6th week of development the embryo will be in an indifferent state. Okay, so this particular anti-Mullerian hormone is also called as Mullerian inhibiting substance. So this particular hormone is responsible for G generation of your Mullerian duct, so that in male there is no formation of female genitalia, whereas in female there will be a presence of Mullerian hormone will be there. Okay, so which are the following contribute to the development of male reproductive system? It is anti-Mullerian hormone. Okay. Next, regarding to this inhibin will be produced by your serotolin cell, which generally inhibit the secretion of FSH and LH from pituitary gland, and GnRH, that is growth hormone, from hypothalamus. It will be having a negative feedback mechanism. Okay. So these 25 questions are highly important. So please pay an attention for these 25 questions that I have uploaded in 2 parts of the video. Thank you friends. Thanks for watching this video.